Also, today's guest speaker is Hannah Karp, so please welcome her.
Sorry, I didn't realize that if I was muted that I therefore could not unmute. So I'll get started. So today I'm going to be presenting on gene editing and talk a little bit about how people have edited and manipulated genomes of living organisms, and then talk about it a little bit in the modern day and focus in particular on CRISPR because I'm from Jennifer Doudna's lab. I work there as a researcher, and I'm sure that a lot of you or all of you are aware that she won the Nobel Prize with her collaborator, Professor Emmanuelle Charpentier, earlier this year. And let's dive into it. So just quick question. Can the students talk to me, or are they all muted?
They are all muted actually, just so there are no distractions, but they are going to be basically sending their questions in the chat box.
Wonderful. Okay, cool. Because I am trying to make this as collaborative as possible. Okay, I'll get started. So here are two drawings of Dr. Jennifer Doudna and Dr. Emmanuelle Charpentier, and they made history earlier this year when the two of them were the first women to win the Nobel Prize. There have been other women throughout history who have won the Nobel Prize, but usually with a male collaborator. One second. So these two women won the Nobel Prize for their contributions to the discovery of the purpose of CRISPR, CRISPR being this mysterious series of genetic sequences. But why is it such a big deal? So what exactly is it that they discovered, and why is it Nobel Prize winning material? So this sort of requires thinking about what it means to genetically engineer an organism and what genetic engineering means just in general. And everybody has a very different mental image when you ask them what genetic engineering is. And so I'd be curious to hear what it means to students in the audience. What do you think of when you think of genetic engineering? I'm going to do the teacher thing where I just sort of wait for.
I guess if someone wants to talk, then just raise your hand and I'll unmute you, if that makes sense. Someone said gene changing or gene changes.
Well, that's almost the definition.
Someone also said genetic modification.
Genetic modification is also genetic engineering.
Yeah, actually, the next person also said genetic engineering could be modifying genes for certain causes, such as medicine.
That's getting closer, but think something specific. Where in your life does genetic engineering play a role?
It's the process of DNA technology after genetic makeup of an organism, changing DNA, creating new species, culture, modifying genes to cure diseases.
Yep, those are all great examples. So that's, but can anybody give me some specific examples? If you think of a particular organism that's been genetically modified, what comes to mind?
Helps make food more accessible by changing its DNA. Mostly foods, humans, cloning. GMO products, mutation animals?
Yeah, again, it's since genetic engineering is a set of tools that can be applied to any living organism, it has a really diverse set of applications. And so all of those are applicable. And I, in particular, have really focused on the therapeutic applications of genetic engineering. And so I wanted to give a little brief history of how people have been manipulating and really engineering genomes for all of human history. So one of the oldest examples is the example of corn, where corn, before anybody could edit it, like Monsanto, wild corn really is this hardly edible, very small plant called, I'm probably going to mispronounce this, teosinte. And it's not tasty, it's hardly edible. And it's been modified over many thousands of years through selection from farmers to be this very large, very yummy, and incredibly resource-rich corn variant that is quite delicious, especially when drenched in butter and salt. And very similarly, people have also been selecting for characteristics that are desirable through breeding in animals. So this is the most generic cow picture that I could find online, but people have been able to select for musculature over the years. So say you want a cow that is able to produce a disproportionately large number of yummy steaks, then you can select for cows that are beefy like this guy. So this is a Belgian blue cow, which is actually a mutation in myostatin that somebody found a cow that just was much, much beefier than all other cows, and then has bred this over the years and maintained this in the population. So really that's an example of genetic manipulation, and by some definitions also an example of genetic engineering. But really what I consider the first examples of purposefully editing the genome really occurred after people discovered X-rays and how X-ray radiation is tied to mutagenesis, so the generation of mutations. And also after World War II, when lots of chemicals that cause random mutations were discovered. And so people began to take common agricultural crops and then just purposefully mutate them. So just blast them with lots and lots of X-rays or chemicals that cause random changes in the DNA. And then just look through many, many thousands of different variants for specific desirable characteristics. So on the left here is white grapefruit. And this is what the plant looked like prior to, I believe, the 1950s. And on the right, you have ruby red grapefruit, which is the most common variant that's sold in supermarkets today. Now, all of the examples that I used previously were based in random mutagenesis and then screening. So pretty much even the example with ruby red grapefruit is something that is naturally occurring by chance. What X-rays do is accelerate the process. So all organisms produce mutations. It's just something that occurs by chance and is actually an error and is usually harmful, which is why getting irradiated is very bad for you and causes often cancer. But the ability to do it purposefully is something that is much more recent. And in the 1970s, a bunch of researchers decided that if they were able to make transgene bacteria, so bacteria that are able to express a protein or a express something from one organism, say a human, but that you could grow in vats and then isolate, that it would revolutionize medicine as we've thought of it. So that's a little bit, that's a lot to think about all at once. So let me use a specific example. Insulin is something that's taken by millions of people in the US in treatment of type 1 or type 2 diabetes. And insulin is not a small molecule. It's a very large molecule that is it's a large peptide. So it's a small protein that's composed of amino acids. And these sequences are the same. So these proteins are produced very similarly in bacteria and in humans and in any organism that's known on the planet. And so what was hypothesized was that you could take the DNA sequence from a person and just put it in to something that called a plasmid, which is a looped DNA sequence, and then you'd be able to grow that protein in bacterium in vats. So previously, diabetics were forced to buy incredibly expensive insulin that had been produced by pigs. So people would go and collect pig pancreases and grind them up and isolate out insulin and then give it to diabetics. And that this was incredibly expensive and also could cause a wide variety of bad side effects because pig insulin is not the same as human insulin. So the people who originally thought of this were very controversial, and nobody really believed that it would work. And so they started with something that was substantially smaller, which is another peptide that is used medicinally called somatostatin. And they took the DNA sequence that encoded for that protein and they cut it into bits, and they weren't sure which region of DNA was what actually encoded for the protein. So they were doing this really in a blind way, but they put it into plasmids and then put it into bacteria and then looked for whether or not that protein was produced. And after several years of troubleshooting this process, they were able to successfully produce these giant vats of bacteria that were all pumping out, like a little factory, the protein that they wanted to isolate that could be used medicinally. And then after a couple of years of modifying this process so that they'd be able to do it for the substantially larger DNA fragment that encodes for insulin, they were able to produce insulin in bacteria, which just blew all of the competition out of the water, because instead of grinding up giant containers full of pig and cow pancreases, instead you were able to isolate it in very, very easily from bacteria in vats. So all of that is well and good, but the editing tools that we can do in bacteria are too invasive and to translate to other systems such as mammals or plants. So it can't be used in the same way therapeutically or agriculturally. So the reason why CRISPR has been such a big deal is because it's allowed us to manipulate genomes very, very precisely, and most importantly, in a way that is programmable. So these gene editors are able to be encoded with a complementary sequence of RNA that can base pair with any region of DNA in a genome as long as you know the sequence. So what this means is that rather than making a series of cuts just by accident and then hoping that one of them produces the characteristics or phenotype that you want and then selecting for it, you instead can say very specifically, oh, I know that this gene makes my cows really, really buff. They can have big, bulky cows if this gene gets activated or deactivated. And then you can go in and cut that. And it can expedite the process of selecting for positive traits to one generation versus thousands, or because you don't have to wait for things to happen by chance. And it also allows for a level of precision that allows for us to apply it in clinical trials in ways that just previously would be impossible. So one powerful example of this is CAR T cell therapy, where CAR T cell therapy is a story of how terminal patients suffering from cancer are able to receive these genetically modified cells that were taken from their own body and then grown up and then put back inside of them. And that these cells have been activated through permanent genetic modifications that then allow them to identify much more readily the tumors, in this case, the diseased, malignant white blood cells in these patients, and then kill them much more effectively and much more selectively than existing treatments for these cancers. So CAR T cell therapy is sounds relatively mysterious, as things often do when they are in acronym form. And the details are not the main point, but what it stands for is chimeric antigen receptor T cell therapy. So chimeric means that you are taking aspects from multiple different organisms or systems and putting them together. Antigen is something that is recognized by your immune system. And receptor is something that is on the surface of a cell that allows for signaling. And if it doesn't make complete sense, that's okay. But what it really means is that scientists are able to put a completely new gene into T cells that are taken from patients to allow them to identify tumors. So basically, every person who has a functioning immune system on the planet has cells in your body, the white blood cells, that are able to identify which cells in your body are you versus not you. And also can identify whether a cell in your body is under attack or is under duress. So if you have a cell that is becoming cancerous or has been infected by viruses, it begins to have things expressed on the surface of the cell that signal to the body that it is under attack and should be killed. So the T cells play this very critical role of identifying the cells that are exhibiting signs of distress and then killing them selectively. But many cancers figure out ways to get around this and stop expressing the key signals on their surface. And so what CAR T therapy does is it pretty much puts in an artificial receptor that allows these cells to, instead of recognizing the normal things that the T cells would look for just during their routine looking about for infected cells, It instead is looking for something that other scientists have determined is expressed on these cells, the tumorous cells, selectively. And then you edit these and grow them up so that there are millions and millions of them. And then you put them back into the patient's body, and then they can go and find tumor cells and selectively kill the cancer cells. And this is just one example of how far genetic manipulation and genetic engineering has come, and that you are able now to reprogram patients' cells within the period of a couple of weeks to allow them to then find and seek out cancers, and just has led to the development of new fields within the last couple of years. So that was a lot, and I kind of meant it to be a lot because I wanted to stimulate discussion, but now I want to open it up to questions.
Yeah, thank you for the information. Yeah, so we do have some questions. I guess the first question I could say was, how easy is it to modify genes?
So it's very easy to modify genes if you want to do what's called a knockout. So there are two different ways to modify genes. Just you can knock out a function where you pretty much take the DNA and you just cut anywhere randomly, and then the body is no longer able to produce that protein. That is pretty easy to do. It is much harder to put in a gene specifically. And really, all of the therapies that involve putting in genes require first taking cells out of patients. So, and it also matters which organism you're trying to edit. So it's much easier to edit some organisms. Say E. coli is very, very easy to edit. Humans are somewhat harder, depends on which cell type, but plants are very, very difficult, is a general rule. Great question.
Yeah, that was a good question. Thanks for the response. Another great question we have is, when you grow more of the edited cells that you mentioned, does the artificial receptor also multiply?
Yes. So the artificial receptor is put into the genome. So it's produced the same way that any protein that would normally be produced in the cells is produced. So it will pretty much, you do the editing, and then you check the, you select for only cells that have successfully been edited, and then you only grow up that pool. So all of the daughter cells also have the exact edits that you looked for.
Yeah, that's a really good response. Another question we have is, related to cancer, is what is the percent of success using this therapy in patients with cancer? I think the therapy that you mentioned, and how popular is the CAR therapy today, and why isn't it encouraged like on radio and chemotherapy?
Okay, so there's a lot of different questions in there. So
Yeah.
one, whenever you're talking about the effectiveness of a therapy, it's very specific to the disease and that particular type of therapy. So for CAR T therapy, it actually depends on which clinical trial you're looking at. The initial small scale clinical trials have been incredibly impressive. So for acute lymphoblastic leukemia, which is primarily a type of cancer that children develop, I think it led to 80 or 90% remission, full remission within a couple of months with but it's very cancer dependent. It's only been used really in cases where the patients would have otherwise been terminal. So only in cases where the patients would have died. And so it's hard to compare it directly to other types of therapies because other types of therapies are used before the patient is so far progressed that they would otherwise die. Whereas these are considered fairly experimental, so they're only used in extreme cases. That being said, they've been incredibly impressive. I would say that if people are interested, they should look at the articles that the New York Times has put out. They're really accessible and well put. Yeah, a major reason why they're not used more widely is one, they're new. Anything that's new needs to be vetted very thoroughly and slowly. And the other main reason is that they are really, really expensive, partially because they're new, but also partially because there's so many steps and so many scientists need to be involved at every stage that it's very expensive.
Thank you. Yeah, I got that question multiple times, so why it's implemented. I think that was a really great response to that. Another question we have is, like, what is used to keep the cells alive after they're taken out of the original organism, and are they edited or grown? How does it work?
Yeah, so there is a whole set of skills called tissue culture where you are able to keep mammalian cells alive independent of, oh man, sorry, the light keeps on turning off because I'm the only one here. Turn on. Sorry about that. Oh, yes, where you are able to keep mammalian cells alive independent of an organism. So they're pretty much in a petri dish. And what you do is you wash them with media, media just referring to this bright pink liquid that contains a bunch of proteins and nutrients and various chemicals that have just been found to keep cells happy. And then every couple of days, you take off that media and then you put on fresh media. And so I babysit cells that just independent of a living organism. But if anybody is really interested in it, I would definitely recommend reading The Immortal Life of Henrietta Lacks, because it talks a lot about how tissue culture was developed and is just a fantastic book. I read it in high school.
Yeah, so that's a great recommendation because many people are high schoolers and students. So I guess building onto that, a question that could help a little people over here is, can you like kind of explain more of your career of how you got where you are, like starting from high school?
Yeah. So I've been a lifelong nerd, so that's nothing new. I knew that I was interested in the sciences really strongly since middle school, but really my entire life. But I didn't quite know what that meant. I knew that I wanted to really have research experiences as early as possible. And I was very lucky that I was in a position to look for them. My high school was decently well connected, and I managed to get a internship my senior year of high school in an oncology lab, so in a cancer research lab at the U of M Twin Cities, so in Minnesota. And after that, I went to university at UW-Madison in Wisconsin. because it had a really good biochemistry program and was relatively affordable for what I was interested in doing, and just has excellent research in general. And then have participated in a bunch of different research labs, really in chemistry and in genetics and in biochemistry labs. And knew that a PhD program was right for me. I guess that there's so trying to fit my whole CV into one paragraph is difficult. But I guess that mostly it's just been a series of somewhat related research experiences that led me to be interested in pursuing biochemistry professionally, and that I applied to UC Berkeley and really had no idea that I would end up working in the Doudna lab. I applied just to the chemistry program as a whole, and then you get to do a series of what are called rotations, which are several week-long internships in a bunch of different labs, and then choose the one that's the best fit. And I love the Doudna lab, so I ended up here. What I currently do is I genetically engineer what are called virus-like particles. So they are similar to viruses. They have the outside parts of viruses, but they don't have any of the DNA or the genome of viruses. And then you can put therapies or something that has therapeutic benefit on the inside, and then it gets delivered to specific cell types or tissues. And we are trying to develop these technologies for gene therapies specifically.
Thank you for the insight. I feel like that's a great response, to give such students interest in working in the same field as you. But building on the virus part you mentioned, someone wanted to know if you are working on the COVID-19 vaccine or tests.
Yes. So the IGI, so me personally, I've just been working on the test kits. So I've been making test kits for just surveillance testing, making sure that the staff and students on UC Berkeley stay healthy and are able to undergo regular testing. The IGI as a whole, so Innovative Genomics Institute in the corner, has been doing a lot of different research activities related to COVID-19 diagnostics and antiviral treatments. So we haven't been working on vaccine development. That's more it requires expansive funds and resources in a way that academic labs don't have. So really places like Pfizer, Roche, Moderna, big biotech and biopharmaceuticals works on vaccine development. What we've been doing is more looking for new antivirals, so treatments for the next pandemic, and also better diagnostics that are portable and easy to use, so that people don't have to wait as long. And the goal that we're really striving for is designing diagnostics that are as easy to use as a pregnancy test, where you can take home a strip and then spit on it and wait a brief period and have a result. It's not there yet, but it's getting, the results have been impressive in the short term.
Thank you. Interesting to know that, interesting projects are going on in the lab. Another question someone had was, if kind of backtracking to the CAR T therapy you mentioned, is what types of cancer can be treated with the CAR T therapy?
That's a great question. At this point, it's exclusively lymphomas and leukemias, so blood-borne cancers. The big barrier in making this more widely applicable is figuring out how to translate this to solid tumors. So there has not been a lot of progress made in applying this to any type of solid tumor. And there are several reasons for that, but primarily it's somewhat logical, where you just, if you think of a solid tumor, these cells have to get in, and also solid tumors create what's called a microenvironment, where they secrete a bunch of different chemicals that make it very difficult for your immune system to identify them. And combating this microenvironment effect has been a very big central barrier. But yeah, it's exclusively limited to blood cancers at the moment.
That's interesting to know. I guess since we have 4 minutes left, kind of like a long question to ask, but kind of backtracking to the internship part you mentioned that you did in your senior year, which is really amazing, is when you were participating in the internship, were you aware of the specific fields you wanted to go, or did it just like help you guide and kind of give you an insight in your career? And if you did know what field you wanted to go into, like, did you focus mainly into that field, or were your options more broad open?
Oh man, I think that when you look at things in post, it seems like I had lots of specific direction, but really that it was a series of relatively random opportunities that I took, and that the clarity and the purpose and the direction came later. And particularly at UW-Madison, it was really, really hard to get a research opportunity because they were really competitive. There were lots of students who were interested in them. And so I took a lot of opportunities just because they were the first ones that were offered to me, and made them work for and incorporated what I wanted. And even the internships that weren't a great match were very clarifying, because I knew what I didn't want to do as much as I knew what I wanted to do, which is a little bit of a cliche, but is true. Frankly, what I think I primarily got used to was just getting rejected a lot, because you ask 20 times and you get a yes once. And that even though it sounds like it was very purposeful, that really a lot of the times it was just what happened to work for me. And that I've been very thankful for all the opportunities that I did get, but that it only makes sense in post.
Yeah, I agree with you. Like, opportunities, as once you get them, you should like take on and run from them. Great response. And then I think before we end at 5:30, an important question that some people had were, what are the risks of CAR T therapy? And like, I guess you kind of talked a little bit about why hospitals aren't implementing it, but kind of more focused on what are the risks upon the body, I think.
Yeah, so that's a great question. I think that's something that a lot of people are becoming more aware of because of this pandemic, is that when you're talking about any type of therapy that involves the immune system, you have to worry about not just what you're fighting, but also the immune system itself. So all of the negative effects are, maybe I shouldn't say all of, most of the negative effects that have been identified from CAR T therapies have been caused by these T cells being overactive and killing off other immune cells that are necessary for vital functions, or causing, it's called a cytokine storm, which pretty much means that these cells can release large quantities of chemicals that send the body into panic. So there have, especially in the earlier trials, some of these patients had complications where their body interpreted these T cells as being, the T cells sort of sent out all of these signals that the body was under attack, and the body began to shut down a lot of major functions, and the complications associated with the rapid activation of the entire immune system can be fatal. So that's part of the reason why they're not broadly applied and won't be broadly applied to anything that's minor. It's in the very least in the near future. Those side effects have gone down with further iterations of these therapies, and are sometimes treated with immunosuppressive regimes. But yeah, the main danger is from the body's response to these T cells, and the T cells can also release these activating signals that can cause the body to panic and attack itself.
Thank you. Yeah, I feel like that was a great response to, I think the question was, it came up multiple times and the response was great. I trust, sadly, we could not get to all the questions, but if one of your questions were not answered, please do email superposition, and we will for sure ask Ms. Hannah for the answers. And otherwise, I think our webinar has come to an end at 5:30. Thank you for the information and the input. I think it was really interesting to learn about genetic engineering. It is like a new thing currently, and it's interesting to learn.
Yeah, it's a little bit of a strange format, so I hope that everybody still got something out of it, but it was a real pleasure, and have a wonderful evening.